Good day, this is Professor Will, CBMD PhD, and today's Saturday, February 29th, 2020. It is 7:14:22 p.m. Eastern Standard Time, and this will be a short, brief, concise, and thorough reading on bioidentification and bioinformatics. So let's get started here. Computerized identification of bacteria is still largely based on the determination of phonetic characters. These include morphological features, growth requirements, and physiological and biochemical activities. Other phenotypic methods include analysis of cell wall composition, cellular fatty acids isoprenoid, quinones, whole cell protein analysis, polyamines, pyrolysis, mass spectrometry, Fourier transformation infrared spectroscopy, and UV resonance Raman spectroscopy. These have not been incorporated into routine computer-based identification systems, although computers are used during the analytic process. Recently, microbiologists have shown more interest in the use of genotypic methods to establish the taxonomic relationships between bacteria, including DNA-based ratio, moles percent, C plus G, DNA hybridization, rRNA homology studies, and DNA-based typing methods. Computers are employed in the collection and analysis of this data, polyphasic identification, and the integration of these various techniques for identification of unknown bacteria. Few, if any, computer-based polyphasic identification systems have been developed. Most computer-based identification systems use only a subset of taxonomic information available to the bacteriologists for a particular group of bacteria. The taxonomy of any group of organisms is based on three sequential stages: classification, nomenclature, and identification. The first two stages are the prime concern of professional taxonomists, but the end product of their studies should be an identification system that is of practical value to others. Therefore, an identification system is clearly dependent on the accuracy and data content of classification schemes and the predictive value of the name assigned to the defined taxa. The ideal identification system should contain the minimum number of features required for a correct diagnosis, which is predictive of the other characters of the taxon identified. However, the minimum number of characters required is dependent on both the practical objectives of the exercise and the clarity of the taxa defined in classification. Thus, many enterobacteria can be identified using relatively few physiological and biochemical tests. The numerous serotypes of Salmonella are recognized by their reactions to specific antisera, and the accurate identification of Streptomyces species requires determination of up to 50 diverse characters. Workers at the Central Public Health Laboratory in the United Kingdom demonstrated the first practical computerized identification system in the 1970s, using a system for enterobacteria. During the same period, the application of computers for numerical classification was developed. This concept was subsequently applied to many bacterial groups, and these studies provided data that were ideal for the development of computerized identification schemes. Many bacterial taxonomists were slow to realize this potential, but these data now form the basis of many probabilistic identification schemes. A wide and increasing range of computerized systems for the identification of bacteria is now reported in the scientific literature and allied to commercial kits. This reflects both the expansion of techniques used to determine characters for the classification of bacteria and the rapid developments in computer technology. The main approach to the identification of unknown bacterium involves determination of its relevant characters and the matching of these with an appropriate database that defines known taxa. This database may be known as a probability matrix or identification matrix. The ideal objective is to assign a name to the unknown that is not only correct but also predictive of some or all of its natural characters. Computerized identification schemes provide a more flexible system than those of sequential systems, e.g., dichotomous keys do. Computerized identification can be achieved in several ways. Numerical codes and probabilistic identification are the most popular approaches. Expert system and neural network have been investigated, but their performance is no better than the probabilistic approach. Numerical codes. These are usually based on plus-minus character reactions. They are applied to a relatively small set of characters that have been selected for their good diagnostic value and are applied to clearly defined taxa. Numerical codes require determination of a series of character states and the conversion of the binary results into a code number that is then accessed against the identification database. Such identification systems are particularly appropriate for the analysis of test results obtained when commercial identification kits are used. An example is the API 20E kit that generates a unique 7-digit number from a battery of 21 tests. The tests are divided into groups of 3, and the results are coded 1, 2, 4 for a positive result for a test in each group. These values are then used to produce a score that reflects the test result, which can be accessed against the identification system. Organisms that generate profile numbers that are not in the identification system can be tested against appropriate computer-assisted probabilistic identification systems. Numerical codes have proved to be convenient and effective, particularly for well-studied groups such as the Enterobacteria ACE. Probabilistic schemes are designed to access the likelihood of an unknown strain identifying to a known taxon. In theoretical terms, the taxa are treated as hyperspheres in attribute space, a space in which the dimensions are the character. The center of the hypersphere taxon is defined by the centroid, the most typical representative, and the critical radius encompasses all the members of each taxon. Ideally, each taxon will be distinct from any others if the identification matrix has been well constructed. To obtain an identification, the diagnostic characters for an unknown strain are determined and its position in the A-space calculators. Thus, in essence, probabilistic identification systems allow for an acceptable number of deviant characters in both the known taxa and the unknown strains. Most computer-assisted identification systems are based on Wilcox's implementation of Bayes' theorem. Classification: orderly arrangement of individuals into units taxa on the basis of similarity. Each unit taxon should be homogeneous and different from all others. Taxon: general term for any taxonomic group, e.g. strain, species, genus. Good day, this is Professor Will, CBMD PhD, and this concludes my reading on bioidentification and bioinformatics. Hope you enjoyed listening. Have a great day.